Hello, everybody. My name is Adelaide Lowe, and I am a third-year undergraduate student majoring in microbiology, immunology, and molecular genetics at the UCLA College of Letters and Science. My research project is done in the Hill Lab, a lab that investigates flagellar motility of the protozoan parasite Trypanosoma brucei. T. brucei is the causative agent of sleeping sickness, a neglected yet chronic tropical disease that is characterized by central nervous system or CNS invasion. CNS invasion reverses the sleep-wake cycle and is life-threatening once neurological changes and motor or sensory problems occur. Over 60 million humans in the sub-Saharan African region are at risk of acquiring infection through the bite of a tsetse fly, the obligate host of Trypanosoma brucei. Livestock is at risk as well, as nagana, the livestock form of sleeping sickness, has become a leading contributor to agricultural poverty in sub-Saharan Africa. And even as a newly or even effective vaccine is being researched, current treatments for sleeping sickness are rendered counterproductive as the rise of newly developing strains and antibiotic resistance remains a concern. The structure that allows for Trypanosoma brucei to be virulent and motile is its flagellum. My postdoctoral advisor, Dr. Daniel Velez Ramirez, previously attributed social motility and behavioral influence to the localization of adenylate cyclase 1, or AC1, to the flagellum tip. How does this occur, and what is social motility? The term social motility, otherwise known as SOMO, defines the communicative behavior that T. brucei exhibits to survive in groups and move towards a desired chemical gradient in the bloodstream of a mammalian host. Now, this red dot that you see here refers to an environmental molecule or ligand that binds to this red receptor. This binding then triggers the conversion of ATP to cyclic AMP, which sends a downstream signal for further elements to be activated for signal transduction. Yet this is mediated by PDEB1, a phosphodiesterase that is required for trypanosomes to move in response to external signals. Now here is another schematic showing you what is happening at the flagellum tip. Once again, you will see the conversion of ATP to cyclic AMP, with cyclic AMP inducing 4 responses. Chemotaxis, which means moving according to a chemical gradient they are attracted to. Social motility, or SOMO, which means that they can communicate with one another once again for transmission, and subsequently disease. Now you see here that this is mediated by adenylate cyclase 1, or AC1. And here the conversion of cyclic AMP back to a different form of ATP is mediated by the phosphodiesterase, of which there are multiple. Now currently there is no known structure of AC1, and understanding the molecular structure will help us understand its function more. Now, first we used AlphaFold to gain some insights into how the protein looks. Now, this is an AI prediction based on AC1's genomic DNA sequence. And we can see that AC1 has 4 distinct domains, including PBP domain 1, PBP domain 2, transmembrane domain, and catalytic domain. We wonder if the structure we obtain will match this prediction. Now here is a proposed linear structure for AC1, and once again, you see all of the components that we discussed earlier. However, my focus is on the 2 periplasmic binding protein-like domains, otherwise known as PBPs. Now, PBPs are bacterial proteins that bind small ligands that regulate chemotaxis. And we believe that the trypanosome PBPs exhibit homology to bacterial PBPs. Thus, their functions are predicted to be similar as well, and that the regulation of adenylate cyclase 1 may be through these PBP domain or PBP-like domains in T. brucei AC1. This will subsequently induce a biological response and subsequent social motility of T. brucei. Now, it has become my research goal over the past year to elucidate the molecular structure of the PBP-like domains and identify its corresponding ligands. Now, some notable methods include Gibson assembly, which is a novel isothermal method to ligating multiple fragments together. We have tandem affinity purification, otherwise known as TAP, to isolate our protein of interest, which is AC1-PBP1, following successful transfection into T. brucei. And lastly, we have microelectron diffraction, or microED, with our collaborator, Dr. Jose Rodriguez at UCLA. Now, after deciding to focus on one PBP at a time, my most recent work is to ligate the tandem affinity purification, or TAP, purification tag, which is called PTP, into the desired vector of interest, PBU82, that contains the PBP1 sequence. Now, the future directions, after identifying both of the PBP domains and their structure, is to determine the structure of the full-length AC1, which is this entire complex right here, and subsequently the identification of its environmental molecular activators. Now, it is my hope that the identification of the binding ligand, along with the 3D reconstruction, will not only be a strong contributor towards vaccine development, but will also allow us to understand how these deadly pathogens interact with their environment using putative ligands or molecules. Now, I would like to extend my deepest appreciation to my PI, Dr. Ken Hill, and my mentor, Dr. Daniel Velez Ramirez, and the other members in the lab who have so strongly supported my growth. I am extremely grateful for the experience and mentorship that I have received throughout my time at UCLA. Please feel free to reach out with any questions or comments, and thank you for your time and attention.